how are you sort of, and you've got an active lab, so.
Yeah, so well, basically, we started out thinking about, well, how is meaning created and how is it processed in our brains, and this is kind of a really hard topic to research because, well, to actually do research on that or to understand how meaning is generated, you basically have to be in a situation where you generate meaning, and this is something we usually do rather automatically or unconsciously. So to actually really investigate the pharmacology of that and the underlying neurobiology, we thought, well, LSD actually is known for changing meaning attribution to our environment. So we thought this would be the perfect tool to actually have people under the influence of a substance where we know that meaning can be changed, and then put them in the brain scanner and make them listen to music and see what happens. And to be able to really dig into the pharmacology of that, we also gave the second drug, which is called Katrin, and which is very selective for serotonin 2A receptors. But in contrast to LSD, it doesn't stimulate the receptor, but it blocks it. So by having these two drugs, we could first look at what is LSD actually doing, how is it altering the attribution of meaning, and then by blocking the serotonin 2A receptor, we could see, well, what exactly is the contribution of this very specific receptor to the alteration. That's what we did.
Okay, and so you had the model, and you were pretty happy with the results from the lab. It worked out.
Yes, we were, well, we were kind of surprised that indeed everything seemed to be dependent on the serotonin 2A receptor, because LSD has a pretty diverse receptor profile. So it does not only stimulate various serotonergic receptors, it also stimulates dopamine receptors. It has been discussed that dopamine, for example, has a contribution as well. So in the end, we were rather surprised that all this generation of meaning seemed to be just dependent on the serotonin 2A receptor. But of course, we were happy about these results because they tell us something new about the role of the serotonin system.
Right. So how important do you think the dopamine receptors are?
So what we saw, and this is also discussed in the paper, but not as prominently because
Right.
it's a little bit more speculative,
Right.
is that the dopamine system might be able to modulate experiences that are already important for us. So what we've seen is a slightly increased brain response with Katrin plus LSD in reaction to in response to the already relevant music pieces.
Right.
So the differentiation might be that serotonin is actually involved in the generation of new meaning, something that has not been meaningful at all to
Oh,
us,
right. Right.
whereas the dopamine system might modulate things that already are
Right.
self-relevant and makes them more or less self-relevant. So this is the thing where we think that there might be a difference between dopamine and serotonin.
Right. So the euphoria that somebody would see in the sort of discovery of new things with LSD, you don't think that's dopamine related? It's probably serotonin related.
That's what we think, yeah.
Okay.
That's what we think. I mean, there might be downstream processes,
Right.
but in general, the first step seems to be the serotonin 2A receptor.
All right. Do you think that's true for other? Okay. So right. So we are generalizing the serotonin receptors as finding meaning in the environment, in stimuli in the environment overall.
Yeah.
Okay.
Yes, and the dopamine system might be responsible, for example, if we encounter an old friend, for example. So
Right.
seeing something that is already meaningful to us, and the rewarding nature of this experience, this might be regulated by dopamine.
So there might be more of a, would it be accurate in a informal sort of way to say that serotonin, you see that you're characterizing the serotonin as more of an exploratory sort of system in a way?
Yeah, that might hold true.
Okay.
I mean, that's not something we can directly relate from the data, but it kind of points in this direction. Yeah.
Talking metaphorically in a sense.
Mhm.
Yeah. Okay. Good. The part of the brain where the serotonin receptors are is now I'm having a sense that it's a strong it's a nice place to look for this sense which is so important in Western culture of identity.
Mhm.
And is that fair? Were you sort of expecting that when you set out? Okay.
Yes, so we kind of knew that well, these areas are important for self-relevant processing. So we were not entirely surprised that it was also these areas which were important for this creation of new meaning. But still, it was interesting to see that these areas which are so important for like coherent self-representation are also the ones which are active and which modulate this attribution of new meaning to things.
And in terms of inputs and outputs, what are these cortical areas? I mean, I'll I'll look it up, but what are they attached to?
I mean, they are rather higher cognitive cortical areas, meaning, so in the cortical hierarchy, they are pretty far up, so they are more like association regions and things. So they are probably related to pretty much a lot of other parts in the brain. So they are probably part of a very large network. Um, so yeah, to find like specific regions, they're related to. I mean, they're certainly related to lower regions, meaning the limbic system, for example. But in general, since they are so far up in the hierarchy, they are probably connected to pretty much the rest of the brain.
Okay, so would you say they're fairly well connected to, say, the amygdala or the basal ganglia or the motor cortices areas, or?
I mean, I would have to look that up.
Yeah.
But I'm pretty sure. I mean, since these are association cortices mainly, they certainly are connected to the limbic system, like the basal ganglia. I'm not 100% sure if there's a direct like link to the amygdala, but there certainly is a link to the amygdala sooner or later. I'm
Okay.
just not 100% sure if it's direct.
Anything on the motor cortices at all? Do you think, or we can look it up?
Oh yeah, I think so. So some of the regions we were talking about are pretty closely related to the motor system,
Okay.
especially the more dorsal parts of these cortical midline structures. They are very close to the motor cortex as well.
Okay, that's interesting. There is a guy in Montreal, Paul Chiswick, who has I think a very coherent model of behavior that's heavily focused on the LIP and the motor cortices, and
Hmm.
it seems like a very compelling model. Where are you going to go next with the research? I mean, what are the problems that are sort of intriguing you next?
Hmm. So, well, in general, for this specific project, we obviously also want to see how generalizable this is. So basically, we want to see does the results we got so far, do they also translate, for example, to other modalities, or is that very specific to music, for example? So can we generalize that to attribution of meaning to, for example, visual stimuli,
Right.
tactile stimuli, something like that? Um, and in general, so I'm fascinated by the question, what is the underlying chemistry of our experiences and behavior? So basically, this is something that I also want to do in the future, and look at other cognitive processes and see how they can be manipulated by certain receptors, or by blocking of receptors, to basically really understand the chemical neuropharmacological basis of our behavior. Because I mean, this is one of the most important things when we think about the development of new drugs, and especially in psychiatry. We haven't had like really new good models. We haven't had really new good drugs for the last almost 50 years. And I think one of the reasons is just because we just don't know so much about the pharmacology of the brain. Um, sometimes it seems like we really don't have any clue where to tap into when we want to develop new medication. And that's why I think it's incredibly important to really look at the pharmacology of the brain and to know which receptor is involved in which experience or behavior. And yeah, and this is obviously a stepwise and iterative process. Yeah, and I'm trying to contribute a little to, well, just acquiring new knowledge in this field.
Yeah, I think that's there. There was an NIH lecture. It was multiple hours of meetings, and there was a representative from one of the drug companies, and she said, "Just give me a target, just give
Yeah.
us a target, please, and then we'll." Are there animal analogies of this? Do you think? And I mean, Can you find rat? I mean, would you be interested in looking at rat or?
So I mean, there are people who are doing the animal research, and I think their work is so incredibly important. So I'm not trained in doing animal research,
Okay.
so I rather stick with humans. But I think it's highly valuable. But then again, there are certain things that we just can't do in animals, like for example, higher cognitive functions like attribution of meaning. I
Right.
mean, that is really, really hard to do in an animal model because you can't ask the animal.
Right.
And this is something that makes it so fascinating for me to work with humans because you can actually have a look into these higher cognitive functions and see what neurochemistry is contributing to these higher cognitive functions that you just can't do research on in animals.
Right. Sort of getting to the last questions, but are you thinking about cross-cultural work at all? Do you think there would be cultural differences at all, or?
Mhm. Um, that is a really interesting question. I have not really considered that so far. I mean, all the studies were conducted in Switzerland,
Right.
and obviously, this kind of research is not super easy to do everywhere in the world. I think we had kind of a heterogeneous group of participants in our study. However, then again, Europe and Switzerland, probably in particular, is maybe not as diverse as in the States, for example.
Right.
So I think it would be very interesting. I don't really think that I'd expect cultural differences, but then again, I don't know. So this is something that would have to be tested.
Okay. Good. All right. Well, I don't have any other questions. I think that's wonderful. And any